Shortly after gastrulation, Drosophila embryos elongate along their anterior-posterior axis as the epithelial cells of the germ-band intercalate with one another in a process called convergent extension. These cell rearrangements depend on the planar polarized localization of actin and myosin II to adherens junctions at the so-called vertical interfaces between anterior and posterior cells, so that actomyosin contractility disassembles these contacts as the cells intercalate. When Sergio de Matos Simoes first joined Jennifer Zallen's lab at Memorial Sloan Kettering Cancer Center in New York, he investigated whether Rho kinase, a key regulator of myosin activity, was also planar polarized during germ-band extension.
And we found that to be the case. Just like actin and myosin, Rho kinase was also asymmetrically localized in intercalating cells, and it was providing an instructive cue for myosin to be at these vertical edges. So this is a paper that we published in 2010. And then the next question was, what's placing this kinase at the vertical edges of the cells? And that's the story of this paper.
To investigate the upstream regulators of Rho kinase, Simoes and colleagues generated a series of deletion mutants and looked at their localization during germ-band extension. Deleting Rho kinase's catalytic domain had no effect on the protein's localization, while removing the C-terminal pH domain mislocalized the kinase to the cytoplasm. The pH domain alone localized evenly around the cell cortex, so Simoes et al. Investigated Rho kinase's central coil-coil domain, which contains binding sites for both the Rho GTPase and the actin-binding protein Shroom.
When we removed the Rho binding domain of Rho kinase, the kinase lost its coordinates in the cell. So rather than being enriched at the vertical adherent junctions, now the kinase was spread throughout the entire lateral membrane and lost its planar polarity. Whereas when we deleted the Shroom binding domain, the kinase was still mainly enriched apically at the adherent junctions, but specifically lost its planar polarity, suggesting that both Rho and Shroom were involved in controlling the planar asymmetry of Rho kinase.
Simoes et al. First looked at the involvement of Rho by injecting embryos with mRNA encoding a dominant negative version of the small GTPase.
The effects on Rho kinase localization were very similar to the ones observed when we removed the Rho binding domain of Rho kinase. That is, again, that rather than be specifically enriched at the junctions of the cells, the kinase would spread throughout the lateral cortex and it lost its planar polarized pattern. Now the kinase would be completely uniform in the plane of the epithelium.
One might expect, therefore, that Rho itself would be planar polarized, but staining for total Rho protein showed no enrichment at vertical adherens junctions. However, the researchers then looked at the pool of active Rho capable of interacting with Rho kinase by expressing a probe containing the kinase's Rho binding and pH domains.
When we overexpressed this construct, we actually observed a small but reproducible enrichment at the vertical edges of the cells. So Rho signaling is slightly, at most, asymmetric in the plane of the epithelium. However, the planar polarity that we observed with this Rho kinase fragment was far from the full-length protein.
However, a Rho kinase fragment containing the Shroom binding domain, in addition to the pH and Rho binding domains, was just as planar polarized as the full-length protein.
So we turned our attention to Shroom, and to our surprise, we found Shroom to be itself a very strongly planarly polarized protein enriched at vertical edges. It's also apically restricted, just like myosin and Rho kinase. So it provided us a very likely target to bias the localization of Rho kinase in this tissue.
Indeed, Rho kinase's planar polarized distribution, as well as its localization to adherens junctions, was reduced in embryos lacking Shroom. Simoes et al. Then examined whether this, in turn, affected myosin's localization and activity.
We performed live imaging of both Shroom mutants and Shroom RNAi embryos. And we observed that in the first 5 to 10 minutes of extension, myosin still becomes enriched at vertical edges. However, from minute 14, 15 onwards, so entering the last stage of germ-band extension, myosin loses its junctional enrichment and becomes spread throughout the entire apical medial cortex when Shroom is absent. So this suggested to us that Shroom was not involved in breaking the symmetry and the initial formation of myosin cables, but it was rather involved in a mechanism that would maintain or amplify the amounts of myosin throughout the second stage of germ-band extension.
At this stage of axis elongation, cells mainly intercalate by forming multicellular rosettes, structures formed by the contraction of robust actomyosin cables spanning multiple cell interfaces.
We observed that rosettes were way more rare in the absence of Shroom, yet more simple rearrangements like neighbor exchange events, which only require the disassembly of a single vertical edge and require less tension, these events were still happening in Shroom mutants. So we were specifically affecting high-order multicellular rosette formation.
Embryos lacking Shroom therefore showed reduced levels of convergent extension. But if Shroom is directing axis elongation by promoting the asymmetric localization of Rho kinase and myosin, how does Shroom itself become planar polarized? Simoes et al. Investigated whether Shroom's localization was dependent on Rho.
And we found that Shroom fails to become planarly polarized when Rho function is downregulated, whereas neither Rho function or localization were affected when we knock down Shroom. So Shroom seems to be acting downstream of Rho activity.
Shroom isn't a direct effector of Rho, but because Shroom is an actin-binding protein, it could be regulated by Rho's effects on the actin cytoskeleton. Indeed, actin failed to preferentially accumulate at the vertical edges of cells in the absence of Rho function.
And when we delete the actin-binding domain of Shroom, we observe that Shroom is completely dispersed throughout the cytoplasm, and only a very small fraction is at the cortex, and that fraction is completely not planarly polarized. So overall, we believe that actin is actually providing an initial cue for Shroom to be recruited to the vertical edges, and this is possibly set up through subtle asymmetry in Rho signaling at vertical edges, with Shroom coming as an amplifying mechanism acting downstream of Rho. Shroom containing a Rho kinase binding domain provides further docking sites for more Rho kinase to be recruited to the vertical edges.
Rho kinase, in turn, can promote Shroom's localization to the vertical edges, possibly through its effects on the actin cytoskeleton. The planar polarization of both actin and Shroom was reduced in Rho kinase mutants, suggesting that these proteins reinforce each other's presence at the appropriate cell junctions.
And this provides a way of phosphorylating more myosin at these vertical edges, allowing the building of strong multicellular cables that result in multicellular rosettes and efficient intercalation. I think it would be important to extend this work to the vertebrate field. For example, it has been shown that Rho kinase and myosin are planarly polarized in the neural tube because the tube needs to extend along its anterior-posterior axis. And structures similar to multicellular rosettes have been described in this tissue. So I think it would be interesting to investigate the possible role of Shroom in the vertebrate neural tube.
Shroom might also organize actomyosin cables at specific cell edges during wound healing or collective cell migration. But you can learn more about how Shroom and Rho direct planar polarized actomyosin contractility during convergent extension in the paper by Simoes et al., published in the February 17, 2014 issue of the Journal of Cell Biology.